The overall goal of our lab is to find better treatments for autism and epilepsy syndromes, two conditions which frequently co-occur. We do this by using animal models of these disorders and by using new cutting-edge tools to look at how the activity patterns of thousands of neurons are altered as animals engage in relevant behaviors. To assist in this autism and epilepsy research, we use new miniaturized microscopy tools, both epifluorescent and two-photon microscopes that we open source and release to everyone in the community, but we can implant these microscopes into the animal models and track the activity patterns of hundreds to thousands of cells simultaneously and try to understand which cell types are actually malfunctioning, for example, when animals engage in social interactions. It gives us a readout of the activity patterns of the cells and actually lets us track these cells over days to weeks so that we can see, for example, how repeated social interactions or learning paradigms can actually affect their activity patterns. We are essentially reading the mind of the animals by recording a large activity in a large subset of cells and then trying to make these activity patterns as close to normal as possible. We feel that having a basic understanding of how large groups of cells synchronize their activity patterns to drive cognition or social interactions is essential for actually being able to understand how these behaviors in essence go wrong in these disorders. So we hope to really at a very fine-grained level understand how cells are connected to each other, how cells communicate with each other, and how these connections and activity patterns become abnormal.